All right, biological macromolecules. I find this super interesting because the molecule's precision or the molecule's structure represents the precision of the universe, almost like there's an intelligent mind behind it. Um, so there's four major macromolecules. There's carbohydrates, nucleic acids, proteins, and lipids. Um, so we'll get into that a little bit, and also why certain things are named certain ways. So probably the easiest one. Oh, before we get into that, we should learn about condensation, which is the same thing as dehydration synthesis, which essentially means that when two molecules bond, one will drop off a hydroxyl group or an OH, and the other molecule will drop off a hydrogen. And by doing so, the hydrogen and hydroxyl group mix to make a water. So whenever two things join together and release water, it's condensation. And whenever two things break apart, it's called hydrolysis. And hydrogen gets put into one molecule while a hydroxyl group gets put into the other molecule. Hydroxyl is just basically like hydroxide, but not an ion. So just OH, essentially. So carbon is an all living thing, super flexible, and is a Lego backbone and framework of molecules. The interesting thing is that for life, we define it as something that has homeostasis, energy use, reproduction, organization, growth, adaptation, and response to a stimuli. So that's not all specific to carbon. Well, it is as we know it. But I'm sure other organisms on different planets, if there are living organisms in the universe, can have those attributes without being carbon based. So, but as we know it, carbon is the only thing that can support life. So, additionally, it's not electron greedy, so it's not too electronegative. Electronegativity basically is the tendency for an atom to rip apart an electron from another. Basically, how bad an atom wants an electron, which is the simplest way to put it. It's defined on the Linus-Pauling scale, but that's in Gen Chem, and this is not Gen Chem. So, what are carbohydrates? So they take the form CH2ON, and they're not just long molecules, they're polymers built from sugars. So we have to define monomers and polymers, and monomers are essentially the building blocks of all four macromolecules. So think of it kind of like a Lego, where it's the simplest part of each of the four major macromolecules. So they take the form CH2ON, so you can think of it as water bonded to a carbon, and a multiple. N is representing multiples. So they're not just long molecules, they're polymers built from sugars. So monosaccharides are simple sugars, and glucose, fructose, and galactose. You can memorize it as FGG, GFG, however way you want to. I think glucose is the most common one. I'm sure everyone knows of glucose. All share the same chemical formula, C6H12O6. Disaccharides are just two sugars linked, pretty common because we have the Greek prefix di there. These are the two macromolecules. Sucrose is glucose plus fructose, lactose is glucose plus galactose, and maltose is just two glucoses mixed together. So polysaccharides are long carbohydrate molecules like starch, glycogen, cellulose, and peptidoglycan. Now, peptidoglycan is its bacteria for bacterial cell walls. So I think the main three that you'll hear a lot is starch, glycogen, and cellulose. So carbohydrates like starch and glycerol are the main energy storage in animals, because animals can digest both starch and glycerol. It's a little bit different for plants because plants only prefer starch because starch is essentially stable. Cellulose is made up of glucose and is made for structural support in plants. The same can be said for peptidoglycan, which are bacterial cell walls, as I mentioned before, that give bacteria shape and protection. It's different from plant cell walls, which we'll get to a little bit later. So carbohydrates are too large to be absorbed, so enzymes have to break them down into monosaccharides. Some enzymes are well, there's amylase, and there is also another one, but I forgot the name of it. Um, so yeah, that's what you need to break it down. So yeah, animals can consume starch, but I'm sure they prefer glycogen. So just know that you have to break down polysaccharides into monosaccharides to get energy. Animals prefer glycogen. Again, it's a short-term energy storage, as mentioned before, and it's stored in liver tissues, and it's easier because we have an enzyme that can digest such carbohydrates. So these are monosaccharides, as I mentioned before, glucose, fructose, and galactose. I don't expect I don't think any teacher expects you to understand the structure too much. So it's a carbon ring, but you can see that there are hydrogens right there that are bonded to hydroxyl groups, hydroxyl groups, hydroxyl groups. So these are just the chemical simple formulas. I won't get into it too much. Nucleic acids are the molecules of inheritance. They're basically genetic information. So they tell cells what proteins to make, what to make them, and how traits are passed on. Nucleic acids are polymers. They're the monomers of nucleic acids are nucleotides, and each nucleotide has three parts. Okay, that is some bad grammar. It should be each nucleotide has three parts. But it's a phosphate group, which is a PO4 minus 3. That should be pretty simple as you learned in chemistry, if you've taken chemistry. A sugar and a nitrogenous base. Now, the two most common nucleic acids that you'll run into, although they're not all of them, are deoxyribonucleic acid and ribonucleic acid, or DNA and RNA, as I've mentioned before. Um, so before we get to that, let's go over the base pairs. For DNA, it's adenine, guanine, cytosine, and thymine, AGCT. You should memorize that. And for RNA, thymine gets replaced with uracil. So it's uracil, adenine, guanine, and cytosine. So the adenine and thymine will always mix for DNA, and adenine and uracil will always mix. And by that logic, you should assume that guanine and adenine are complementary base pairs. Two things to note is that we'll get into this a little bit deeper because it doesn't make any sense for now because you don't understand how the bonding works. But just know it's hydrogen bonding, and we'll get to it a little bit later there. There's also something called epigenetics, which I've been looking into, but that's completely out of the scope of this class. So DNA and RNA structure. DNA is double-stranded, and its sugar is deoxyribose. So remember, a nucleotide is made up of a sugar, a nitrogenous base, and a phosphate group. So the sugar used for DNA is deoxyribose. So notice that everything for for why can't I remember? So everything, I'm just gonna say DNA. Um, Everything for DNA is just something with the DO or dia approach or deoxy behind it. Um, so for RNA, you can just remove the deoxy and get ribose. The same thing is over here. Wait, hold on. Why am I blanking right now? I forgot the name of DNA's long name. Oh, it's deoxyribonucleic acid. Yeah, it's pretty simple. So you could just take out the deoxy there. So that's how to memorize it if you need that. The double-strandedness is important because it protects genetic information and prevents the base pairs from bonding. But guess what? We get to transcription RNA and messenger RNA all later. But RNA is single-stranded because, again, it stores genetic information, and we're going to need it to replicate certain things. So that's why it's single pair, and that's why it's easier to bond because it's not protected from an outside shell, much like DNA is. So first of all, I think that's amazing because how did our bodies even do that? That is just pretty amazing. So, um, there are 20 amino acids. Technically, there are 22. I think there's something called selenecysteine. Um, let me. Yeah, it's selenecysteine and some other one. But they're not really used. 20 are the first 20 are the main ones. I believe the 21 and 22 were discovered recently, so those aren't really that important. These are the monomers of proteins. So an amino acid consists of a carbon, a carboxyl group on the right side, and a side chain, and an amino group on the left side. Amino group is NH2. Carboxyl group is minus COOH. Note that the hydrogen at the end can actually fall off and protonate the substance, which essentially means to drop a hydrogen ion or proton into the substance, which is why it's called an amino acid. The R group is what makes each amino group different. The R group is the same thing as the side chain. The R group can bond to other R groups, and the main functions are enzyme, structure, transport, protein signaling, and defense. I'm sure you know because protein speeds up recovery if you're an athlete. Central carbon is in both groups and connects both. The interesting thing about the central carbon is it contributes to both of the structures of each molecule, which is interesting. Polypeptides are the same thing as every other macromolecule we've learned so far. They're just groups of the same monomers. So they're made up of one or more amino acids via peptide bond. A peptide bond is pretty simple. It's just when the amino group bonds with the carboxyl group. So remember that the carboxyl group is on the right side, and I have a picture on the next slide, I think. Actually, I don't. So I guess I'll just show you in there. So fortunately, I don't think you'll have to memorize the structure, but it's good to know because um, I know it's not that hard to uh it's not that hard um to memorize. C and then NH2. Well, actually, it should be like this. Sorry, I don't know why I'm blanking right now. Let's go back. Um, so, um, so remember the carboxyl group is on the right side and the amino group is on the left side. And if you know your geometry, actually it should be well, this should be up here. If you know your geometry, well, you don't even need geometry for it. You can use logic. Um, Typically one is double bonded, as you see here, and we have the NH2 on the right side. So this is a side chain, and there's nothing on top usually. Oh wait, there is, because it's a hydrocarbon, obviously. So there is a hydrogen on top, I believe, yeah. So what happens is if another R group comes and bonds with this one, um, then we get a I don't think they call it this, but I guess it would be called a dipeptide. So we get R, C, H, and then again the amino and the carboxyl group. But we can get to the carboxyl group and we can get even further bonds with the amino group. So since the amino group is on the left side, another molecule uh, can. I'm rusty. I forgot. Okay, there we go. That's the right. Can come and bond with this carboxyl group. So this amino group right here and this carboxyl group, or actually it would be this entire thing, can bond together, and that's what a polypeptide is, which is essentially what a protein is. The primary structure is like writing a sentence. So this is the structures of protein. I don't think they're too important right now because we do get into the building of protein later. Um, so what happens is in the primary structure, it's essentially long linkages of amino acids to make polypeptides. So again, polypeptides are just mixing up amino acids. So it would essentially be um, amino. So think of amino as words, and think of this entire thing as sentences, which is essentially what's happening. Then what happens is the secondary structure is the local folding of it. So what will happen is proteins will bind themselves together using intermolecular forces, which is essentially attractive electrostatic attractions, and they'll bend into certain shapes. But we'll also learn that a little bit later as well. And the tertiary structure is the full 3D structure caused by R group interactions. Remember, the R group is the side chain. Something interesting about proteins is they can actually denaturate, which is essentially or denature, which is essentially when the protein is under immense stress and it starts to unwind. Which is why I believe when you cook eggs, you can actually denature the proteins inside the eggs. So that's pretty interesting if you didn't know that already. So two types of protein folding structures are alpha helices, which are coiled spiral structures. Think of it like a spring, but it's not exactly like a spring. It looks like a spring. Beta plated is a folded sheet like structure. Alpha helix is usually a little less stable as the beta plated. So that's pretty interesting to know. Neighboring strands are held by hydrogen bonds, like we mentioned before. In fact, you should always know that before we continue. You should always know that intermolecular forces are what holds molecules together. Pretty simple stuff. Lipids are hydrophobic. So this is the last macromolecule. And hydrophobic just means water repelling molecules. Now there are lipids that essentially act like a detergent, where they have a hydrophobic end and a hydrophilic end. Um, so yeah, so they are so they're just hydrophobic molecules. They are mostly made up of carbon and hydrogen, so they're not really um, oh shoot, I should not have said that. Are not true molecules. They're definitely molecules. I think that was a typo right there. But the main roles are energy storage, membrane, and protection. So there's four types. Triglycerides, which are essentially fats and oils. Fats and oils have 9 calories or 38 kilojoules of energy. I don't know if you will need that, but maybe you would need that for a nutrition course. Phospholipids are essentially the hydrophobic end and the hydrophilic end, which means water-loving end and a water non-loving end. Steroids are four fused carbon rings, and waxes are its own special type of molecule that's actually used in coating things to make it waterproof. So that's interesting. Um, Triglycerides are made up of one glycerol plus three fatty acids. Glycerols are essentially three carbons bonded together. Hydrogens, three carbons bonded to three hydroxyl groups. Three hydrocarbons bonded to three hydroxyl groups. So yeah, or actually, a chain of a three carbon chain with three hydroxyl groups. That's, I guess, the best way to put it. It does insulation and cushion protection of organs. Again, they're fats. So yeah. Saturated fats have no double bonds, but unsaturated fats do have double bonds, making it liquid at room temperature. Fatty acids are long hydrocarbon chains with a carboxyl group at the end. Carboxyl groups, again, can be acidic, um, um, which is why, uh, which is why proteins are called amino acids, or the monomers of proteins are called amino acids. So this is glycerol. As I've said, that they have the hydroxyl groups at the end, and the hydroxyl groups bond to three fatty acids. And they're called fatty acids because, again, they have a carboxyl chain that bonds with the hydroxyl groups. Interesting stuff, right? Phospholipids are made of glycerol plus two fatty acids. So now we take out a fatty acid and we add a phosphate group, PO4 minus 3. It is amphiphatic, which means it has both hydrophobic and hydrophilic regions. Now, the reason I find this so fascinating is there's something called the phospholipid bilayer, where essentially two phospholipids make it so that the hydrophobic end is on the inside. So that means that it can still bring water inside the molecule through selective permeability. So that's how cells get water, through their cell membranes. Steroids are essentially lipids with four fused carbon rings, as I've mentioned before. They are for hormones, cell signaling, and membrane structure. Now, people think steroids are a bad thing, but everyone has cholesterol, and we eat cholesterol, so um, that's one steroid. So don't think of steroids as some foreign molecule or foreign group of atoms. They're just something that regulates membrane fluidity, and it actually stabilizes animal cells. But too much can actually lead to something where if the cholesterol bonds to the blood vessels, they can cause a rupture if the vessels are weak, and also they can build up in your system, causing blood clots, if you didn't know that either. So this is what a carbon ring is. You can see these carbon rings right here. So carbon, 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 and there's four of them. So yeah, these are actually, I believe, for resonance, but we won't have to focus on that for now. Passive versus active transport. So this slide is kind of ordered weird. The active transport should go below the facilitated transport. Passive transport is movement across a protein channel that does not require ATP. So no energy is required. And this is because low to high diffusion, or high to low diffusion, high to low concentration. So a concentration gradient is essentially how much how the concentration moves along a water system. So high to low concentration is pretty obvious. You drop a piece of salt into water, the salt's not going to stay there. It's going to disperse because all the salt is concentrated into that drop of salt that you dropped into the water, but it wants to disperse because that's the statistic tendency of it. Like think of spending money at a grocery store. Your money is not going to sit there. It's going to want to spread. So that's diffusion. And that's basically the basis of entropy, which is actually really important in active transport. Now, active transport is movement across a membrane that does require adenosine triphosphate. It moves substances from low to high concentration. But no, earlier we said that everything wants to move to high to low concentration. Well, this is where entropy comes in. So notice before that naturally things go from a tendency of high to low concentration, but you can actually unnaturally change things from low to high concentration. So we have pumps, endocytosis, and exocytosis. So endocytosis is when things enter the cell, exo is when they leave. But before we do that, we have to learn about entropy. Well, we don't have to learn about entropy. So as we said, that dollar or that $20 you spend at the grocery store is likely not going to sit there. It's going to go through the cash register. It's going to get turned into change. It's going to disperse. But interestingly enough, if you put enough energy and time into making sure that $20 doesn't disperse, then it won't. It's the same thing here. You can move things from low concentration to high concentration if you put enough energy in through the protein channels. Um, so entropy states that things move from high to low concentration essentially. That things go from that things disperse. Concentrated energy tends to disperse. So it goes from a low to high concentration, um, or it goes from high to low concentration. But here you can do the opposite by pushing energy. Think of it like putting all your clothes. Let's say you have a closet and you put everything in your house into that closet and you close the door. Sure, everything is messy inside. You moved all the outside stuff into a really concentrated area. Um, but the natural thing it wants to do is that when you open that door, everything's gonna fly out at you and it's gonna hit you and it's gonna fall all over the floor. But that door is essentially that energy just keeping everything inside. So that's what active transport is. Again, facilitated transport should have been above, but it's simply the movement across the cell using transport proteins without using energy. So again, it's passive transport. Just know the difference between passive and active transport. Endocytosis and exocytosis. Um, Endocytosis is the movement of materials into the cell using energy. It forms a vesicle and is used for large particles or fluids. So endocytosis is just inside, exo is outside. You should know that if you've taken chemistry. Endothermic is absorbing heat, exothermic is releasing heat, stuff like that. So there's two types. There's phagocytosis for solids or large particles, I believe, and pinocytosis is for fluids. Um, so I'm sure you can think of a way to memorize those two, but I think it's pretty simple. Exocytosis is, again, movement out of the cells using energy, ATP. So both use energy, and the vesicle fuses with the cell membrane. So that's essentially what exocytosis is. You don't really have to know that second part. That's just kind of an example. So yeah, why? Oh, I don't know why I put the waxes at the end. They're long fat. Wait, waxes? Yeah, long chain fatty acids. So again, fatty acids are just long hydrocarbon chains with a carboxyl group at the end. So they are for waterproofing and protection, such as beeswax or plant leaf coating. Oh, actually, they're also linked to alcohol too. Oh yeah, linked to alcohol. So, um, I think that's it. So, yeah, they more specifically they're esters of long chain fatty acids and long chain alcohols. But again, this is an introduction, and that's biology, macromolecules.